This is my video for Module 4 on how to make a wet mount for EDIT 5380 in the summer of 2015. The purpose in this video is in teaching the correct technique in making a wet mount, and the audience is really for any biology student, but primarily for high school through college. And the difference between a wet mount and a prepared mount is that a wet mount is very fast, it's quick, it's temporary, it's easy to do, it is a staining technique, and it utilizes a cover slip. A prepared mount is permanent, it's much more time consuming, a whole lot more technique involved in it, so it's not as easy, but it is also stained and it uses a cover slip, but that is cemented down, and then it will have a permanently affixed label to it. The materials that you need obviously are going to be slides and cover slips, lens paper, toothpicks for the demonstration that I'm going to show, and some staining method, and in this instance we will use methylene blue. By the time we get all said and done, we will view it under a microscope, and I pulled out my old field microscope. It's an oldie but a goodie, but in my lab I have a $70,000 microscope, and wet mounts work just as well on it as well. The objective here is for students to make as well as stain their own wet mounts, and the true test is going to be making sure that they don't have any bubbles, and then viewing their wet mounts through the microscope. So the end result, they should see something in the realm for epithelial cells with the cheek smear that we're going to do is the nucleus, the cytoplasm, the cell membrane at different magnifications, and it should stain easily and using good technique. Hi, I'm going to demonstrate on how to make a wet mount with beautiful technique. First thing, you need to start with a glass slide, and you need to dry polish it to get any fingerprints or smudges off of it using lens paper, which is the only type of paper we use on cleaning slides. So I'm getting this all nice and cleaned up, and you notice I'm holding it by the edges to keep from getting any fingerprints on it. Now, another good technique to use is to use a China marker and basically draw a little outline for where you plan to put your specimen. And if you can see that, I don't know yet, we'll see. And the technique that I'm demonstrating today is using a cheek smear to make your wet mount. So I've got a toothpick, and I'm going to gently scrape the inside lining of my cheek. All we're doing is picking up a few cells, and then we're going to gently smear them around on the slide, get them nice and spread out. And then the next thing to do is take 1 drop of methylene blue to use as our stain. Just 1 drop right there in the center. And then we take our cover slip, just a little teeny glass square, if you don't drop it. And then the most important part here is to hold the cover slip at a 45-degree angle to the slide, touch it to the slide, drag it until it touches the stain, and then drop it, and there you have a beautiful temporary wet mount with no bubbles in it that you are ready to view using your microscope, starting out on the low power magnification, working up to higher and higher magnifications. The nice thing about the microscopes in the labs is that they are parfocal, and you start out on the low powered objective, switch to the next higher powered objective, and it should be in focus or just nearly so. But the best part about it is no drips, no runs, no bubbles. Beautiful wet mount every time. Thank you. I hope this was helpful.